Hello, my name is Marina Fandaros, and I'm a third-year PhD student in Dr. Wei Yin's lab. For my BME Research Day talk, I will be presenting fluid structure interaction studies of patient-specific coronary artery disease biomechanics. Coronary artery disease is the leading cause of death in the United States. And as plaque builds up in the arteries that supply blood to the heart, it decreases the blood supply, as shown in the cross-section, and poses risk for heart attack, shown in the bottom section. These plaques can be composed of calcium, lipid, or a mix of both. Our research focus is to be able to develop a model of the main coronary arteries from a healthy patient, and we can then create models for unique cases that each have a different plaque composition, either calcified, non-calcified, or mixed, as shown in these representative images. The coronary artery can be identified as branching off the base of the aorta and can be extracted from CT scans to be reconstructed in 3D. We modeled the left anterior descending coronary artery, which is here highlighted in green, since it supplies blood to the left ventricle, which then supplies blood to the rest of the body. Our model will incorporate the blood flow and arterial wall to look at the fluid flow and the biomechanics. This would include the fluid velocity, wall shear stress, and wall strains. And by analyzing our models in this way, we can understand if there's a specific hemodynamic pattern that would be particular to the plaque type. And as we can see in these representative geometries, there's a large variation between the patients and their plaque types. So we have a healthy model, calcified, mixed, and non-calcified. This is a plot of the fluid velocity in the artery over the cardiac cycle, speeding up at the area that is narrower, indicated between the two blue arrows, to 0.7 meters per second at the inflow when it's the highest. We can also analyze the wall shear stress that shows a normal range between 0 and 12 pascals, again increasing the most at the area of narrowing. Overall, we're able to take patient images, extract the geometry, and perform fluid structure interaction computational analyses to quantify the biomechanical parameters. Our future work will consist of making more patient models and understanding how clinicians can better use this type of analysis to quantify the biomechanical environment and the plaque to understand what the plaque experiences. Ultimately, this can be used as a predictive model to non-invasively sort an unknown case to the correct plaque type. Thanks so much for listening to my talk. For questions, please contact myself or my PI, Dr. Wei Yin, whose contact is on the screen.